This video clip presents the summary of research project entitled Compressive Force Upregulates Notch Target Genes and NOTCH2 mRNA in Human Dental Pulp Cells. Dental pulp can encounter direct compressive force when the tooth moves, such as during trauma, mastication, or orthodontic treatment. Dental pulp cells can also encounter hydrostatic compressive force during pulpal inflammation, which can be induced by various stimuli, for example tooth preparation, orthodontic forces, dental caries, trauma, and parafunctional habits. Mechanical forces can induce biological response of dental pulp, including cell proliferation, adhesion, migration, osteogenic, and odontogenic differentiation. The response of dental pulp cells to mechanical forces was reported to involve many signaling pathways. In this study, we focused on Notch signaling pathway in dental pulp cells subjected to mechanical compressive force. Notch signaling is activated in a juxtacrine manner upon binding of Notch ligand to its receptor on a neighboring cell. In mammals, 4 Notch receptors, NOTCH1, NOTCH2, NOTCH3, and NOTCH4, have been identified. Binding of Notch receptor to its ligand induces 2 consecutive cleavages of Notch receptor, resulting in the release of Notch intracellular domain, which further translocates into the nucleus and eventually activates Notch target genes. The well-known Notch target genes are Hes and He family genes. In this study, we hypothesized that compressive force increases mRNA expression of Notch target genes and Notch receptors in human dental pulp cells. To prove the hypothesis, we performed an in vitro experiment using primary human dental pulp cells obtained from non-carious third molars of healthy donors by cell explant method. The cells were subjected to compressive force by 2 compressive force application models, direct compression and hydrostatic compression. The mRNA expression of Notch target genes and Notch receptor was then investigated by real-time polymerase chain reaction. From the study, we found that both 1 and 2 gram per square centimeter of force from both direct compression and hydrostatic compression models can upregulate Hes1 mRNA expression at 2 hours after force application. However, upregulation of He1 was not observed. Next, we investigated the effect of force duration on expression of Notch target genes. We found that at longer duration of force application, which was 6 hours in this experiment, hydrostatic force could stimulate Hes1 and also stimulated He1 mRNA expression, while upregulation of Notch target genes was no longer observed in direct compression model. For Notch receptors, the only change in expression that we observed was an increase in expression of NOTCH2 following 6 hours of hydrostatic compressive force application. In conclusion, both hydrostatic and direct compressive forces can upregulate the mRNA expression of Notch target gene Hes1 in human dental pulp cells. However, hydrostatic force produced more prolonged activation of Hes1, and it also stimulated the upregulation of He1 as well as NOTCH2 at longer duration of force application. The results from this study could serve as the basic information for further studies that will lead to better understanding of molecular mechanisms occurring when dental pulp cells encounter mechanical force. This knowledge could be further applied to enhance the repair process of dental pulp and reducing the destructive processes, including dental root resorption, following the clinical situations such as tooth trauma, parafunctional habits, and orthodontic tooth movement. Thank you for watching our video.